HIV. Its full form is Human Immunodeficiency Virus, which is causative agent of AIDS. Okay, and around 40 million people all over the world are suffering from this HIV, and every year around 2 million people die due to this, and 1.5 million new cases are coming every year. Okay, now it belongs to family Retroviridae and subgroup Lentivirus. Retro means reverse. It means that there is central dogma of biology according to which DNA is from DNA there is formation of mRNA, and from mRNA formation of protein. But in this case there is gene of this virus is RNA, which is reverse transcribed into DNA, and from DNA there is formation of mRNA and genomic RNA, and there will be formation of new virus. Here one enzyme is working, that is reverse transcriptase enzyme. Lentivirus means its latent period or incubation period is around, it takes around 7 to 8 years. Means if there is penetration of virus today in human body, then clinical symptom will appear after around 7 or 8 years. Okay, now this virus is single stranded, this virus of single stranded RNA, which is dimer means it has one pair of RNA, means same identical RNA. It has capsid, which is icosahedral. Icosahedral means it have 20 sides. This virus is spherical and enveloped because it is present in lipid bilayer membrane. Now on its membrane, which is lipid bilayer, it have protein, that is GP41 and GP120, and inside this inside this structure there is capsid, which have on capsid there is protein P24, and here is RNA, one pair of RNA, and inside capsid there is RIP, means enzyme, reverse transcriptase, integrase and protease. Now this virus attach with our those cells which have only CD4 positive receptor. CD4 positive receptors. CD4 positive receptors are present on T helper cell, monocytes, macrophages, microglial cell, skin dendritic cell, which is also called Langerhans cell. In spleen and lymph node there is follicular cell. These all cells have CD4 positive receptor. Now precursor of GP120 and GP41 is protein GP160. GP160 is precursor of this protein, means GP120 and GP41. Now how this virus attach with our macrophage? On macrophage there is CD4 positive receptor, and this GP120 it attach with CD4 positive receptor, and this is called primary attachment. And then it after primary attachment, two more side of this GP120 activated, and it attaches with co-receptor of macrophage, which is CCR5, means chemokine receptor 5, and co-receptor of T helper cell is CXCR5. Now primary attachment and secondary attachment done. After that GP41 it hook with the membrane of macrophage, and it pull this macrophage around it, and there will be fusion of this virus membrane and macrophage membrane. In this way it enters into macrophage body. Now how transcription happen? There is a single stranded RNA. From single stranded RNA, first single stranded DNA is formed, and enzyme work is reverse transcriptase, and from single stranded DNA double stranded DNA formed, and from double stranded DNA mRNA and protein is formed. Now function of reverse transcriptase is it helps in formation of single stranded DNA, and once single stranded DNA form, then it catabolizes the single stranded RNA. Now function of integrase enzyme, it helps it cut the human DNA. Suppose this is human DNA, and it cut our human DNA and then add this DNA into human DNA. And once this process is done, there is no any drug up to now is formed which can destroy this DNA. Now about gene. There are 3 genes: GAG, ENV and POL, Pol gene. What is function of GAG gene? GAG gene helps in formation of cell and capsid. Means this cell and capsid. On capsid there is P24 protein. Now ENV, function of ENV gene, it helps in formation of transmembrane protein which is GP41 and GP120. GP120 it is also called spike, and GP41 it is transmembrane protein. Now function of Pol gene, it helps in formation of RIP, means enzyme, reverse transcriptase, integrase and protease. Now about drugs. There are 4 drugs. First one is called attachment inhibitor, means it inhibit the attachment of GP120 with CD4 receptor. Name of this drug is Maraviroc. Now second drug is fusion inhibitor, means it inhibit the GP41, means it block the GP41 so that fusion of GP41 with macrophage membrane is inhibited, and the name of drug is Infuvertide. Now third drug is NRTI, means nucleotide reverse transcriptase inhibitor. In this what happen, we give pseudo nucleotide with the orally, means with tablets, and it reverse transcriptase enzyme helps in making new DNA with the help of this pseudo nucleotide, and this process is not working, and name of this drug is lamivudine. Now fourth drug is NNRTI, means non-nucleotide reverse transcriptase inhibitor. Means this drug, what do function of this drug is to inhibit the reverse transcriptase enzyme, means it inhibit the reverse transcriptase enzyme. Now test, there are 2 tests, ELISA test and Western blot test. We detect the protein P24 in the body with these tests. First there will be detection of P24, then we find other proteins like GP41 and GP120 with the help of these tests.